everyone. I'm PJ Brooks. I'm the Deputy Director of the Division of Rare Disease Research Innovation at NCATS, the National Center for Advancing Translational Sciences, a part of the National Institutes of Health, and also one of the co-chairs of the Bespoke Gene Therapy Consortium, or the BGTC. And we all know that gene therapy holds promise for treating rare and genetic diseases, but the journey from the lab to the patient often depends on something people don't see or think about, which is actually manufacturing gene therapies. So in this community quick take, developed in partnership with the BGTC, we will explore the complex behind-the-scenes process of making adeno-associated virus, or AAV, gene therapies and why it matters to the patient community. And indeed, we're doing this session in part in response to questions from BGT members and patient advocates. So I'm very pleased to be joined by Dr. Stephen Gray, a professor in the Department of Pediatrics at the University of Texas Southwestern Medical Center, and the co-director of the Gene Therapy Program and Viral Vector Facility there, and also Angelica Meyer, the Director of Manufacturing Science and Technology at Aldebaran. So let's dive right in. Maybe this first one I'll start with Angelica. If you can address what is a plasmid and how does it help in making an AAV gene therapy?
Yeah, absolutely. So bacteria have a very unique structure within them called a plasmid. Although bacteria have a standard DNA within their cells, similar to humans, bacteria have a really unique extra part of their DNA that's called a plasmid. What these plasmids allow bacteria to do is quickly and easily communicate really critical information. So if a bacteria learns about an antibiotic, and it develops its own resistance, it can very easily transmit that information to other bacteria. And therefore, that trait can very quickly be adopted by a generational group of bacteria. Because of the quickness of their ability to transmit this information, this allows bacteria to make plasmids very readily and rapidly. And the AAVs or other genetic treatments or other genetic materials or RNA or proteins can be coded by scientists within these plasmids. And so what that means is that we can take a bacteria that is coded with these plasmids, that has it engineered already in the cells, grow them up, and have produced the bacteria plasmid in a large amount. And so the quantity is where it's really important. So if a bacteria has a plasmid that has been transmitted for what is needed to make the AAVs, we can grow this much quicker as opposed to doing a molecule by molecule synthesis. And so from the making of the plasmids, that's when they would go to somebody like Dr. Gray to take them to the next step for an AAV therapy.
Great. So Steve, can you expand on then how you use the plasmids in making AAV?
Yeah, and I think maybe a simple way to think about it is, the plasmids are just going to be the pieces of DNA that have the instructions that we would put in a producer cell to make the AAV. So we're basically, taking some producer cell, we're introducing different pieces of DNA that will make the different components of the virus and the gene that we want to package in the virus. And all of that is coded on the plasmids. So these are raw materials that we use to make any of these AAV vectors being used in gene therapy.
Great. Okay. Second question then is, why are quality checks so critical in making gene therapies? And what kinds of things do scientists look for to make sure a therapy is safe and works as intended?
I'll just chime in on the bacteria or on the plasmid part, because the plasmids have to be very clean and very correct in order for Steve to do the magic that he needs to do. It's really critical as we are extracting these from bacteria that there aren't any residual bacteria parts in there, as any extra bacterial material could cause the expression that Steve does to be incorrect, or even worse, to cause a negative response in the host through the presence of any bacterial material that could cause an immune response. So it's very, very critical that we not only are making the correct plasmids to make sure that they code correctly for what is needed, but also that they do not have any extra bacterial debris or bacterial machinery or any other byproducts of bacteria.
Yeah, and so I'll just follow up on that really quick. Even the context of like making AAV, just remember this is a very complex biological drug. It's an engineered virus. We're putting in an artificial piece of DNA into it. We're producing it in cells that can carry, and we're using chemical processes to kind of purify that that gene therapy drug. And so all those steps along the way, you want to make sure you're always manufacturing the right thing, because facilities can be manufacturing multiple different products. You want to make sure that one of them doesn't contaminate the other. And just think about if you're if you're going to the drugstore and you're getting a pill or something that you're taking, or you're some kind of injectable, you want to make sure that it's safe. You want to make sure that everything in it is what you think it is. You want to make sure that that it has the potency, that something hasn't happened where it got heated up and left too long somewhere, and and you're putting it in your body and it's not working. So all of this, when you talk about quality checks, it's a complex manufacturing process. There's all these raw materials that come in, and all of those raw materials, it's just like you can have garbage in, garbage out. So you want to make sure that all the things that you're putting in are really high quality. You're checking and double checking to make sure that they are high quality. And then along the way, you want to make sure that your manufacturing process is consistent, that you're making the same drug at the same quality, the same purity every time. Because ultimately, these are things that we're putting into patients, we're putting into young children, um, and especially if these are experimental medicines that are going into clinical trials, you don't want a situation where maybe you see adverse effects and you're wondering, is this because of the drug, or is this because of some contaminant that got in it when it was being manufactured, and something wasn't done right. So we don't want a potentially really good drug to kind of get halted and sidelined because there was a mistake that happened in the manufacturing. That's all these are really critical therapeutics that are moving forward, and we have to make sure that when we do go into patients, it's being done responsibly, and that the results that we see are really because of the drug and not because of some other problem. Some other technical problem.
Okay. So next question is, maybe this one is for you, Steve. Why is it hard to make enough gene therapy or AAV gene therapy for everyone who needs it?
So, and a lot of this we're talking about, say, systemic diseases, like muscle diseases, brain diseases, liver diseases, where we're trying to manufacture, like you have an engineered virus that's trying to get into the cells in the body. And basically how well that works is dependent on how many cells actually take up and receive the gene. So you have to end up, pushing doses pretty high. And right now, the field of AAV gene therapy is really, I think clinical trials were starting about 25 years ago. Some of the first clinical trials around using AAV, was about 25 years ago. So it seems like a long time, but there's been a lot of evolutions in the technology, and a lot of those evolutions have come in the manufacturing. And we're kind of at a point for a long time where we could barely manufacture enough of the drug. The knowledge around manufacturing those types of AAV gene therapies, the knowledge was very specialized. The methods for manufacturing them were complicated. There were a lot of things that could go wrong. And in recent years, a lot of those things have become more standardized, but it's a complex biological drug. And and so when you're scaling up to manufacture, maybe you can manufacture enough to treat 10 patients, and then you want to treat 100 patients. Well, you might have like a 50-liter tank that can, of, manufacturing bioreactor that can manufacture enough for like 10 patients. And then you say, all right, well, I want 100 patients. Now that's 500 liters. Okay, I want 1,000 patients. Now that's 5,000 liters. That's a big tank. And and that's really how things have gone when people are trying to treat things like Duchenne muscular dystrophy, where the doses that being administered to patients are massive, and there's thousands of patients out there that, tens of thousands of patients out there that need the treatment. So scaling things up, it's just a logistical nightmare, and then all the raw materials that are needed are very, very expensive. It's a huge logistical problem. And the solution to that is to make the manufacturing processes more efficient, to make it more standardized, um, to hit economies of scale. And I think that that's kind of where everything's going. And I think that's one of the big ambitions of the Bespoke Gene Therapy Consortium. Um, but in the meantime, we're kind of in a situation where we are having to make like sort of these relatively large batches to treat a relatively few number of patients. And so they're expensive and they take time. And it needs to get better.
Yeah, and within the BGTC, we're working with sort of the standard types of AAVs and trying to reuse these, realizing that, of course, people are developing newer and better ones all the time. So another question that comes up a lot from patients in particular, and all of us, is a question of time. How long does it take, and why does it take so long? And so maybe I'll ask you and ask that about how long does it usually take to make a batch of plasmid that someone like Steve can then use to make the AAV, and then how long does it usually take to make a batch of AAV vector? So Angelica, how about you start with how long does it take to make the plasmid?
Yeah, so as Steve mentioned, it's really critical that what we make is completely traceable. So he needs everything that's going into his manufacturer to be very clean and very traceable. So do we, even though we're, ahead of that, all of the materials that come into Aldevra need to be traceable. The process needs to be very controlled. And so making the material at a high GMP level for utilization in AAV for patients although it only takes a couple of weeks to make, there's all of the critical documentation aspects and traceability aspects of materials. All of our materials have to be single use so that we are not introducing any kind of unknown contaminants. As Steve mentioned, some of the negative ramifications of that. And then once we grow the bacteria and then purify the bacteria for each individual plasmid, there's a substantial amount of work on the backside of actually making the plasmids to ensure that they are pure, that they do not have any extracellular or extra materials from the bacteria, to make sure that they are exactly what they need to be, and then to make sure all the traceability and the documentation is in order. And so that whole entire process, again, although it takes about 2 weeks to make the plasmid, it can be months to go from start to finish of a batch, because of the documentation, the materials, traceability, release documentation, etc., to ensure that everything is up to where it needs to be, the level of cleanliness, prior to then going on to the next step for AAV manufacture.
Wow, so it could actually take longer to do the documentation and testing of the plasmid than it does to actually make the plasmid?
100%. Yeah. And so again, it's super critical that it's clean and pure, of course. But that's absolutely the most critical aspect is ensuring and controlling the quality of the materials that leave our facility.
Great. And then Steve, why don't you address the same one with regard to actually making the AAV?
Yeah, I mean it's this is rinse and repeat. This is the same story because except the plasmid is one of the raw materials that comes into it, but everything else there's um it might take a month to grow up all the cells that you're going to use to make the AAV. And then, actually making the AAV could just take a couple of weeks. But leading up to that, you've got prep work to uh, sort of model the manufacturing at a small scale, making sure that the AAV is producing the way that it's supposed to, making sure that you're going to get the kind of yields that you need to, making sure that you're not getting any surprises in terms of it not packaging correctly or um, not having potency issues. So you kind of have to do some of these small scale productions up front. And if you don't do that and you just go into a large scale manufacturer, then if that fails, that's really bad. It's a very, very expensive failure and it sets you back even further. So you've got to do these sort of small scale pilot productions up front. You want to make sure that the materials you're using for those are also well controlled, high quality. And then, as you're doing those small scale manufacturing up front, then, it sounds silly, but when we talk about the quality controls, you've got to test the product to make sure that the purity is good, to make sure that the potency is good, to make sure that, it's suitable to go into a human being. Those experiments, those like analytical tests that have to be done, those take some work to develop and make sure that they're generating consistent, reliable results from, just that analytical method works. So then that's like some lead up activities before you're even ready to start manufacturing of the large product, that all of that has to be in place. And then sometimes, some of these raw materials that you're using could have, a 3 month or a 6 month lead time, to acquire those. So, and then, after you manufacture the drug, then it can easily be 2 or 3 months of time to complete all the release testing, and to have all the documentation and the reviews and the quality oversight to make sure that all of those analytical tests and quality controls were done properly, and that drug is really ready to release, to a human patient. So, again, you're talking about, you start to add those things up, and it could easily be, 6 months, 9 months, a year longer, kind of going from start to releasing a product. Easily. And then some of the manufacturing facilities, they're not just sitting with an open queue twiddling their thumbs. They've got batches lined up for different clients. And so when you come to them, they might say, "Oh yeah, we can make this for you, but our queue is full until we get about 6 months out, and then we can start on it." Um, and I know on the patient side and on the clinician side and on everybody's side, everybody's pulling their hair out because they want it to go faster. And they'll say, "Well, it's just paperwork. Can't you just get the paperwork done?" But, the reality is these are complex manufacturing processes. There's a lot of things that are really easy to do wrong. And if you don't have these controls in place, then things are going to go wrong. And then you're either going to have to stop and redo things and take longer, or you're going to make a product that could harm a patient instead of do good. So it's very, very frustrating, but there are reasons, and this is normal.
Great. Well, I think you've both done a really nice job explaining, what is involved in making AAV, starting with the plasmid, how long that takes, and how a lot of that is really the testing even more than the manufacturing, and ultimately those timelines do impact getting into a clinical trial, but of course are essential to be sure that the material that goes into that patient is safe and effective. So I think that's it for us. Thank you both very much for taking the time to talk to us today.